Good day everyone, I'm here again to answer the self-testing question in genetics. Self-testing question number 1. Consider three pairs of homologous chromosomes with centromere labeled A over A and B over B and C over C, where the slash line separates one chromosome from its homologue. How many different kinds of meiotic products can this individual produce? My answer for question number 1 is 8. Question number 2. There are 56 chromosomes in the somatic cells of channel catfish. Question letter A. How many chromosomes does a channel catfish receive from the male parent? My answer is 28. Letter B. How many autosomes are present in channel catfish gamete? Letter B, my answer is 56. Letter C of question number 2. How many sex chromosomes are in channel catfish egg? My answer is 2. Letter D of question number 2. How many autosomes are in the somatic cells of a female parent? My answer is 28. Question number 3 in self-testing 1. In rainbow trout, cells are diploid, having 28 pairs of chromosomes. How many of each of the following are present in each cell at the stage of mitosis and meiosis? Letter A for question number 3, centromeres at prophase. Letter A for question number 3, my answer is 48 female and 50 male. My answer for letter B, the chromatins and anaphase, 0 female and 2 male. Sternopsis diapana is known to have an XO sex determining system if it has 24 pairs of autosomes. Letter A for question number 4. How many chromosomes are in a female and male Sternopteryx? My answer is 48 female and 50 male. Letter B. How many sex chromosomes are in the female and male Sternopteryx diapana? My answer is 0 female and 2 male. Self-testing number 2, question number 1. Differentiate DNA from RNA. RNA contains the sugar ribose while DNA contains the slightly different sugar deoxyribose. And RNA has the nucleobase uracil while DNA contains thymine. Question number 2. Differentiate transcription from translation. Transcription is the process of copying a gene's DNA sequence to make an RNA molecule, and translation is the process in which proteins are synthesized after the process of transcription of DNA to RNA in the cell's nucleus. Question number 3. How do DNA replicate? It replicates in the major steps: the opening of the double helix and separation of the DNA strands, the priming of the template strand, and the assembly of the new DNA segment. Question number 4. Discuss how our proteins are made. Proteins are built as chains of amino acids, which then fold into unique three-dimensional shapes. Bonding within proteins molecules helps stabilize their structure, and the final folded forms of protein are well adapted for their function. Self-testing number 3, question number 1. A dominant G allele is common car-produced normal pigmentation, and the recessive G allele or small g allele products gold pigmentation. G allele produces gold pigmentation. A cross between normally pigmented and gold common car produces all normal progenies. Letter A from question number 1. What are the genotypes of the parents? My answer is each parent contributes one allele to each of its offspring. Each parent contributes one allele to each of its offspring. Thus, in this cross, all offspring will have the BB genotype. Letter B. What are the genotypes of the progenies? An offspring genotype is the result of the combination of genes in the sex cells or gametes, or sperm and ovary, that came together in its conception. One sex cells came from each parent. Sex cells normally only have one copy of the genes for each trait, one copy of the Y or G form of the gene in the examples. Letter C. What genotype should be muted to produce an old gold phenotype? My answer is red feather phenotype, dominant and recessive alleles. The other allele is usually recessive, which means it is only expressed if there are no alternate forms of the gene, other alleles present. Therefore, both the genotypes AA and small a produce a red feather phenotype. Question number 2. Based on the theory discussed on the hybrid cross, all the gametes produced by a hybrid cross of two homogeneous gray guppies with curved spine. Letter A. The gray guppies with a curved spine will be produced 16:0 ratios. And letter B. Genotype ratio 16 GG and CU 16/0. Phenotypic ratio 16 gray guppies with curved spines is 16. Question number 3. Based on the given example on X-linked genes, how a transparent tailed guppy is produced? Prove your answer to A. Draw a Punnett square and determine the phenotypic and genotypic ratios of the progenies of the cross between the mating of the heterogeneous caudalist female and transparent-tailed male. My answer for letter A question. Self-testing number 4, question number 1. For example, if a calf farmer wants to select for increased weight at 18 months of age, for decreased dressing percentage, or based on a head size to body size ratio, and for increased gain per day, he assigned relative values to the three phenotypes. The following are the values that he assigned for his project. Phenotype: population average relative importance, weight 350 grams, 40%. Phenotype: dressing percentage, population average 65%, relative importance 30%. Phenotype: gain over day, population average 2.5 gram, relative importance 300%. Explain the result obtained. What do you mean by your value? The answer in question number 1. Question number 2. A three-breed cross is used to produce various combinations of dominance genetic variants and hybridization. For example, a farmer wants to produce a fast-growing at the same time cold, tolerant tilapia in the slightly saline water. Using breed O aureus, A.O. niloticus and O. Zambicus M, design a breeding program that will give you 50% and 35 and 37.5% A and 12.5% M. Illustrate your answer. Was the farmer able to accomplish his goal? Explain. Question number 3. For example, say a hatchery manager manager spawns and raises Thai catfish, native catfish, and their reciprocal hybrids in the other to evaluate the relative growth of these catfishes at his hatchery. He harvested the four groups when they are 12 months old and records the following average weight. The question is, determine the mean weights of the parental groups. Letter B, calculate the mean weight of the hybrids. Letter C, calculate heterosis. Letter D, explain the result. Self-testing number 6. Discuss briefly the different ways of producing monosex male population in tilapia. It is originally achieved by normal sex reversal of newly hatched fry using mainly methyl and testosterone added to feed. Number 2. Enumerate and discuss the things to consider for a successful sex reversal treatment. First is sex reversal of fishes must begin before the gonadal tissue of young genetic females has differentiated into ovaries. Second, depending on water temperature. Large numbers of free-swimming fry can first be found in warm water pans about 2 to 3 weeks after stocking. Next, fry production must be coordinated with the sex reversal operation because the hormone treatment should be begin immediately after fry harvest. Question number 3. Briefly discuss how electrophoresis is undertaken and how it is used to describe the population. Electrophoresis is a laboratory technique used to separate DNA, RNA, or protein molecules based on their size and electrical charge. And that's all. Thank you.